This video is brought to you by Saraswati Vidyalaya Union. And hi, this is Prapti. Today we are going to study glycolysis from the chapter 13, Respiration from standard 11th. Now let's see in detail.
The next step that we are going to study, that is about the aerobic respiration. In the aerobic respiration, 3 steps are there. First one, that is known as the glycolysis. Second one, it is known as the Krebs cycle, and third one, it is known as the ETS. Now let's start this aerobic reaction. In that, the first step that is known as the glycolysis. So this glycolysis, which is common in aerobic respiration as well as anaerobic respiration, because for the glycolysis the oxygen is not required. So that's why this is the common state which occurs in the aerobic respiration as well as the anaerobic respiration. Now remember this one, this glycolysis, it is also known as the EMP pathway. In the examination, either by this name the question can be asked you or by this name, EMP pathway. Now why it is known as the EMP pathway? Because here what happened, this glycolysis state which is discovered by the 3 scientists. That scientist name, that is Emden, Meyerhoff, and Parnas. So these scientists discovered this pathway. That's why this pathway, by their name, this pathway which is known as the EMP pathway. Okay, so the thing is what? This glycolysis. Glycolysis means what? So breaking of glucose in systematic, by the particular reaction or the enzymatic breakdown of glucose into the 2 molecules of pyruvic acid. Enzymatic breakdown of glucose into the 2 molecules of pyruvic acid, that is known as the glycolysis. Glyco means glucose, lysis is breaking down. Okay, so that's here, this glucose which is broken down into the 2 pyruvic acid, 2 molecules of pyruvic acid. That's why this process is known as the glycolysis. Okay, this glycolysis process which occurs in the cytoplasm of cell. Where? In cytoplasm of cell. That's why this glycolysis, it is also known as the cytoplasmic reaction. Okay, now in this glycolysis, if you observe here, this glycolysis completed in the 2 steps. Okay, the first one, that is known as the preparatory phase. Preparatory and cleavage. Okay, and the second one, which is known as the oxidation and payoff phase. Oxidation and payoff phase. So in these 2 phases, this glycolysis process completed. Now in this glycolysis, if you observe here, this is the schematic representation of glycolysis. See that purposely here I have shown different colors and here I have shown different colors. Now in this glycolysis there are total 10 steps. You can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Total this process of glycolysis is completed into the 10 steps. Now here from 1st to 5, this till this here till this 1, 2, 3, 4, 5. Okay, so that first 5 steps are there which are known as a preparatory phase and cleavage. Then the remaining reactions which comes into this oxidation and payoff phase. Okay, now here what happened? Here the first step, you can various steps occur. If you observe here, that is known as the phosphorylation. Then here second one, isomerization, again phosphorylation. Then here, that is known as the isomerization. Here, here cleavage. Then isomerization. Here oxidative dephosphorylation. Then here dephosphorylation. Then here the process that is known as the isomerization. Then dehydration and then dephosphorylation. All these are the steps completed into the glycolysis. Now let's see first step. That first step is what? That is known as the phosphorylation. Means this glucose molecule undergoes phosphorylation in presence of ATP, as well as there is one of the factor is present that is known as the magnesium ion factor is present. So in presence of that magnesium ion and the enzyme that is known as the hexokinase enzyme. So in presence of that, glucose convert into the glucose 6-phosphate. Because the process is what? Phosphorylation. Phosphorylation means what? Addition of phosphate. Okay, so that glucose molecule converts into the glucose 6-phosphate. In the starting, that phosphate group is attached to the first carbon compound in the glucose, but afterward it comes toward the 6th carbon compound. So that's why it is known as glucose 6-phosphate. Okay, that first step that is known as the phosphorylation. Then the second step, which is over here, this second step that is known as the isomerization. Means this glucose 6-phosphate isomerized in presence of enzyme that is known as the phosphoglucoisomerase, or you can say that phosphohexoisomerase. So this glucose 6-phosphate get isomerized in presence of this enzyme into the fructose 6-phosphate. Okay. Then the next step which occurs, the next step that is known as the phosphorylation. Again what happened? That fructose 6-phosphate in presence of ATP and the enzyme that is known as the phosphofructokinase. So it undergoes phosphorylation and it forms the fructose 1,6-diphosphate. Okay, the product is what? Fructose 1,6-diphosphate. Then here what happened? The process is of cleavage. Cleavage means what? This one molecule which get divides into the two molecules, that is known as the cleavage. So here this fructose 1,6-phosphate, it divides into the two molecules that is known as the glyceraldehyde 3-phosphate, and the second one that is known as the dihydroxyacetone phosphate in presence of enzyme that is known as the aldolase enzyme. Okay, so here glyceraldehyde 3-phosphate, it is also known as the phosphoglyceraldehyde. This is also known as the phosphoglyceraldehyde. Then after the cleavage, so remember this one, these are interconvertible, means here the dihydroxyphosphate can convert into the phosphoglyceraldehyde, or the phosphoglyceraldehyde can convert into the dihydroxyphosphate. Okay, that interconversion takes place into that. Then this phosphoglyceraldehyde undergoes isomerization in presence of enzyme that is known as a triose phosphate isomerase, and then it forms a glyceraldehyde 3-phosphate. It forms a one molecule only that is known as a glyceraldehyde 3-phosphate. Means that is phosphoglyceraldehyde only. Okay, then here that phosphoglyceraldehyde 3-phosphate, it undergoes the next stage that is known as the oxidation. Okay, during the oxidation, so this oxidation process takes place in presence of this NAD. Okay, so before starting the further process, remember this one, till this process, it is known as a preparatory and cleavage. Okay, and the remaining one that is known as the oxidation and payoff phase. Okay, like that here, that preparatory phase is first to 5 undergoes oxidation in presence of that NAD, as well as see here in presence of this, there is the addition of H3PO4 takes place. This process completed in presence of triose phosphate dehydrogenase. Okay, so in presence of that glycerol dihydrophosphate convert into the 1,3 biphosphoglycerate. In shortcut you can write down like this: 1,3 biphosphoglycerate, or you can say that phosphoglyceric acid. Okay, so then this 1,3 biphosphoglycerate, it undergoes dephosphorylation. What is given? 1,3 biphosphoglycerate. It undergoes dephosphorylation and then it forms a 3-phosphoglycerate. यहाँ पे bi means 2 phosphate groups are there. From that 2 phosphate group, so there is a dephosphorylation takes place in presence of the enzyme that is known as a phosphoglyceric kinase enzyme. So that one phosphate group removed from this and it forms the 3-phosphoglycerate. Okay, so here what happened here? There is a production of ATP dextrose because that is a process of dephosphorylation. Okay, means from that phosphate, from that phosphoglyceric acid only that phosphate group is removed. Then this 3-phosphoglycerate undergoes isomerization in presence of enzyme that is known as a phosphoglycerate mutase enzyme, and then it forms the 2-phosphoglycerate. Okay, then again the next step that is known as the dehydration. Dehydration means what? Removal of water substance, water molecule. Now here see that this 2-phosphoglycerate undergoes dehydration in presence of enzyme that is known as the enolase enzyme, and then it forms the 2-phosphoenol pyruvate. Okay, then at the end here the 2-phosphoenol pyruvate means what? In short you can say that 2-phosphoenol pyruvate is what? Pyruvic acid. That means it is known as the PEPA. Okay, that is phosphoenol pyruvic acid. Then again it undergoes dephosphorylation and at the end it produces the pyruvic acid. Okay, and here the enzyme which is required, that is known as the pyruvate kinase enzyme. So in presence of this enzyme, in presence of this ADP, so there is a formation of pyruvic acid. So like this is the process is of glycolysis. Once again in short I will explain the steps. First one, phosphorylation. Second one, isomerization. Again phosphorylation, then cleavage, then isomerization, then here oxidation, then dephosphorylation, here isomerization, dehydration, here dephosphorylation, and at the end, so from one glucose molecule there is a formation of 2 pyruvic acid. So this is the systematic enzymatic breakdown of glucose into the 2 pyruvic acid, and that's why this step is known as the glycolysis.
Thanks for watching this video. Please like, share and comment this video, and please subscribe my channel. My channel name is Prapti Vanzare.